Hi and welcome to ExtraSci. This is Life Sciences Grade 10. We're still in Module 1 looking at life at a molecular, cellular and tissue level. Now we move over to Unit 3 looking at cell division through mitosis. Under this unit we'll be looking at chromosomes, the cell cycle and then cancer. Let's start off with chromosomes. So what are chromosomes? Chromosomes are thread-like structures found in a nucleus. They transfer genetic or hereditary information from parent to offspring. They consist of DNA wrapped around proteins, and before cell division, the chromosomes are kept in a chromatin network. This chromatin network is a mass of threads that shorten and thicken, forming chromosomes. Under this section, we'll be looking at the basis of division and then DNA replication. Cells are multicellular, which means they have many parts and are able to divide continuously. The cell division of a somatic or a body cell resulting in growth is known as mitosis. Mitosis is when the mother cell divides into two daughter cells that are similar to one another in the mother cell. And before cell division happens, DNA replication has to occur. So before mitosis happens, DNA replication has to occur. And during mitosis, the mother cell, which is one mother cell, divides into two daughter cells which are similar to one another in the mother cell. Now when this happens, as you saw in the video, there was one cell which divided into two other cells. So before there was one cell and after there were two cells. Not that before there was one cell and then after there are three cells. The original cell is not there at the end of division. DNA replication. This is the process happening before cell division. DNA and chromosomes. DNA replication is a process by which DNA makes exact copies of itself. Replication occurs such that the daughter cells have the same genetic material. It was said that mitosis is when the mother cell divides into two daughter cells that are similar to one another in the mother cell. Now this is possible because during DNA replication, the DNA replicates itself such that the daughter cells have the same genetic material. So the mother cell had a strand of DNA. The DNA replicated and now there were two strands of DNA. And when the mother cell divides into two daughter cells, each daughter cell has one strand of DNA which is the same as the other daughter cell, which is also the same as that of the mother cell. And after replication, the chromatids in the chromatin network have also doubled, and they join to form double-stranded chromosomes joined by a centromere. As you can see these threads here looking like cotton, these are the chromatin network. The chromatin network will double and then shorten, thicken, forming chromosomes. These are our double-stranded chromosomes which are joined here at the centromere. The centromeres are the centers of the double-stranded chromosomes. Now the cell cycle. The cell cycle is the period during which the cells grow, replicate, which also means copy. So it's where they grow, copy their genetic material, and divide. By genetic material, we mean DNA. There are two types of cell division, mitosis and meiosis, but we'll only be looking at mitosis for grade 10. You will look at meiosis once you reach grade 12. Mitosis is the division of the somatic or body cell. Meiosis is the division of gametes or sex cells. Before every cell division, the cell goes through interphase. Interphase is the process that occurs during DNA replication. As I did say, DNA replication happens before cell division, and interphase is where DNA replication happens. So interphase also happens before cell division. So we have interphase, and during interphase DNA replication happens. And after interphase we have mitosis, or after interphase we have meiosis. Under this section we'll be looking at the process of mitosis, then we'll look at the role of mitosis. The process of mitosis, the major phases. Mitosis occurs in two stages. Stage 1 is karyokinesis, and then there is cytokinesis. Karyokinesis is the division of the nucleus and the chromosomes. Cytokinesis is the division of the cytoplasm and the cell membrane. It has four phases. There is prophase, metaphase, anaphase, and telophase. So prophase, metaphase, and anaphase fall under karyokinesis, and telophase falls under cytokinesis. So during prophase, metaphase, and anaphase, we are dividing the nucleus and the chromosome, and then during telophase, the division of the cytoplasm and the cell membrane is occurring. Now let's look at that in more detail. Starting off at prophase, this is a diagrammatic representation of what happens during prophase. As you see, we already have chromosomes here. So the chromatin network has already formed chromosome. And notice what is happening here. This thing is disappearing, and you can see that this thing is starting to form. So let's read that information. The chromatin network condenses, shortens, and thickens, forming chromosome. I've said it an awful lot of times. You are now familiar with the chromatin network forming chromosomes. As you can see, the chromosomes are forming during prophase. The nuclear membrane and nucleolus disappear. As you can see, the nuclear membrane here going around and around, it's starting to disappear. And then the spindle fibers begin to form. These things up here are called spindle fibers, and they are only beginning to form during prophase. So what is happening during prophase is preparation. It's preparation for mitosis. The cell is preparing itself to divide. So a quick way to remember prophase is to think of it as the preparation phase, where the chromosomes form, the nuclear membrane and the nucleolus disappear, and the spindle fibers start to form in preparation for division. And then after preparation, we have the middle eclipse, metaphase. So looking at the diagram first, you can see that your spindle fibers have now joined up like this, and they have moved all throughout the cell, and you can see that your chromosomes are in a straight line at the center. Now let's look at our points. The chromosomes move to the equator and arrange themselves in a row along the equator. They move to the center and arrange themselves along the center. And the spindle fibers attach themselves to the centromeres. The centromeres are the chromosome centers. So as you can see, the spindle fibers have attached to the centromeres here. They have attached to the centromeres. They've all attached to the chromosome centers. And our chromosomes have moved to the equator and are in an eclipse. An eclipse is when one thing is directly in front of another. As you can see, all of the chromosomes are directly in front of another in a row along the equator. Middle eclipse. They have arranged in the middle at an eclipse. This is what happens during metaphase. So before we had preparation, which was prophase, and now the middle eclipse, arrangement in a row at the equator, which is our metaphase. And now we move on to our third phase, which is anaphase, apart navigation. As you can see, they are navigating apart. Your chromosomes were at the center. They are starting to move apart. And you can see the spindle fibers are also detaching from each other. Each spindle fiber is pulling the chromatids to different poles. As you can see here, it says the centromeres of the chromosomes divide into two. So you can see